Doctors. Today we are going to see Neet Neurolog series day 8. Successfully we have completed 1 week of code words. Before starting, I am going to ask you a question. Okay? What are the reticular structures seen in a cell? If you know the answer, type it in the comment section. Next, can you name any two important code words used by the TA to ask for plasmid, to ask about plasmid? Okay, so what are plasmids? Do you know what are plasmids? What are the special characteristics of plasmids? It is very important in the prokaryotic cell as well as in the class 12 biotechnology lesson. So can you name any two important characteristics of plasmid which is used by the NTA code word in the NEET MCQ exam? If you know the answer, quickly type it in the comment section. Now, as we planned yesterday, our long-term goal is to clear NEET and be a medico. But the short-term goal is to focus right now here for the 30 minutes, only 30 minutes. Okay, shall we start? Are you ready to lock everything in your gray cells? Students, shall we start now? The first block is about scientist and discovery. Okay, scientist and discovery. Already we have seen about Rudolf Virchow, Schleiden and Schwann. Then who discovered the living cell? Now the question is, who first discovered the living cell? If you know the answer, type it in the comment section. Next we studied about Robert Brown, Fleming, and today we are going to see the rest of the scientist and discovery in the cell chapter. First, Golgi apparatus, discovered in 1898 by the scientist Camillo Golgi. Okay, if you minus 67, this year is of importance, very important. We already saw. If you know the answer, type it in the comment section quickly. Golgi apparatus. These are reticular structures. These are reticular structures. Okay, seen close to the nucleus. They are seen close to the nucleus. These 2 points are important. Usually scientist and discovery will be asked only as match the following. Sometimes even if they match the following, there will be a twist. They can ask as reticular structures close to the nucleus. Okay, so these 2 points you have to remember in NEET point of view. Shall we start memorizing it? Golgi apparatus, discovered by Camillo Golgi in the year 1898. Camillo Golgi. Did you find out the answer for this question? 1898 minus 67, 1831. Who discovered nucleus? It is Robert Brown. We already saw N for N. Okay, N for nucleus. Now again, Golgi apparatus, 1898 by Camillo Golgi. Golgi apparatus, they can ask you Golgi apparatus or reticular structures close to the nuclei. They are seen close to the nucleus in the year 1898 by the scientist Camillo Golgi. Okay. Next is the ribosome. What are these structures? Ribosomes are granular structures. You know what is the meaning of granules? Granular structures like mustard. Have you seen mustard? It's available as a spice in the kitchen. Granular, like this, granules. Okay, sugar granules. We can see sugar granules. So granular structure. These are granular structures seen by George Pallade in the year 1953. Discovered by George Pallade in the year 1953. So either they can ask you as ribosomes, match the following George Pallade, or with the year, or they can give it as Pallade particles and match it with the scientist, that is George Pallade. Okay, so these ribosomes are named as Pallade particles after George Pallade. ribosomes are granular structures identified, that is discovered in the year 1953 by the scientist George Palade. So after him, ribosomes are named as Palade particles. Ribosomes. Now there is a question for you. What are the types of ribosomes seen in a prokaryotic cell and eukaryotic cell? If you know the answer, quickly type it in the comment section. The ribosomes in the prokaryotic cell is of what type and eukaryotic cell is of what type? 70S versus 80S, isn't it? Ribosomes, they are granular structures discovered by George Palade. Ribosomes are of two units. Name the smaller and the larger unit. 50S and 30S is equal to how many? We saw already, we shouldn't add it. It is a sedimentation coefficient. So it is 70S, which is seen in a prokaryote. Okay, and 80S is seen in a eukaryotic cell. Ribosomes are granular structures discovered by the scientist George Palade. Okay, they are like sugar granules. Attached to what? Now there is one more question I am going to ask you from the previous class. If ribosomes are attached to the endoplasmic reticulum, they are called as rough endoplasmic reticulum. If there is no ribosomes, it is called a smooth endoplasmic reticulum. Now what is the function of RER and SCR? If you know the answer, type it in the comment section. We already finished this. Next, ribosomes are also called as Palade particles. They are also called as Palade particles after the scientist George Palade. Okay, Palade particles. Second time, ribosomes are also called as Palade particles. This is very important. What is the function of ribosome? What is the function of ribosome? If you know the answer, type it in the comment section. Ribosomes are also called as Palade particles. Now see, they can give the match the following. They can give the organelle, straight away the organelle, or the structural appearance of the organelle, or the other name of the organelle, and match with the year or with the scientist. Okay, this is a mix and match like this. Next, the fourth one is the plasma membrane or cell membrane. Very, very important, and very, very important in all point of view. That is match, statement type, AR type, as a direct question. That is, who proposed the plasma cell membrane model, fluid mosaic model? They can ask it as a direct question. And AR type. Okay, the question here is about lipids and proteins. Now before going into this, I am going to ask you whether it is lipid base on which proteins float, or the proteins C on which the lipids float. Now choose any one. If you know the answer, type it in the comment section, A or B. I want to test you whether C. There is an ocean. Okay, you can see the icebergs floating on the ocean. Okay, these are icebergs. One is the lipid here, and the floating one is the another option. You have to choose which one is lipid and which one is protein. I have given two options. Okay, if you know the answer, type it in the comment section. Now coming back to this plasma membrane or cell membrane, what is the important characteristic of plasma membrane or cell membrane? It acts as the doorway to the cell, a gateway, doorway to the cell. This is the one who has the right to choose. This is the one which has the right to choose who has to go inside the cell and come out of the cell. So you call that characteristic as what? Selective permeability. It is like you are inside the class and you are asking, whom will you ask the permission? Your ma'am. So ma'am has to decide who has to come in and who has to go out. So selective permeability. Okay, this is very important, seen both in the prokaryotic and eukaryotic cell. A common characteristic. See, everything you have to study, which means you have to understand the concepts. This is one of the similarity between the prokaryote and eukaryotic cell. Now, plasma membrane or cell is discovered. The fluid model is proposed in the year 1972 by Singer and Nicholson. Okay, so plasma membrane or cell membrane model. What is the name of the model? Fluid mosaic model. Fluid mosaic model, or the quasi fluid nature, quasi fluid membrane. This model is proposed by Singer and Nicholson. This one we have tried 3 times definitely. Plasma membrane, also called a cell membrane. In 1972, the nature of the cell membrane is found to be a fluid mosaic model or quasi nature. They can ask in either way. Okay, they can mention it as fluid mosaic model or quasi fluid membrane by Singer and Nicholson. Next, third time, plasma membrane, also called a cell membrane, which is selectively permeable. In 1972, Singer and Nicholson proposed the model of cell membrane, that is fluid mosaic model or quasi fluid membrane. This is very interesting to read. We'll study it in detail in the video classes. Now I said there is a phospholipid bilayer. Phospholipid bilayer. You have to remember very easy here, the lipid and the protein are the heroes. Okay, so there is an ocean. I told you, imagine an ocean with icebergs floating on the ocean. They'll go like here, there, because they are free to move, right? But here there is lateral movement. Lateral means what? Side movement, side to side movement. So in the ocean of lipids, these proteins are like floating icebergs. They can move laterally like this. Nature they call it as fluidity. So one of the important nature of the plasma membrane is fluidity of the plasma membrane, which has got many functions. Okay, so shall we move to the next? Students, what are the important characteristics you remember about prokaryote? Okay, mention that in the comments. Type it in the comment section quickly if you remember any one important characteristic of prokaryote which is related to the Neet exam. They are used as a code word by the NTA. If you know that, type it in the comment section. So now we have finished about 4 scientists and discovery. Now moving to the prokaryotic cell. P for plasmid. P for plasmid. Okay, so when you remember, it is very easy. Plasmids are seen in prokaryotic cell, particularly a bacterial cell. Okay, plasmids are seen in prokaryotic cell, particularly in a bacterial cell. What are the code words used by the NTA so that the answer should be plasmid? We are going to see it. See. Plasmid MCQ. The code words used by NTA are extra chromosomal DNA. If you see a word extra chromosomal DNA in the question, then the answer is plasmid. Then the second question, second code word, autonomous replication. What is ORI? Origin of replication. Okay. So before going into this, I missed to tell you this one. Okay, in this plasmid, you have to, when you are studying plasmid, you have to divide it into 2 parts. Okay, one is of bacterial importance, which is given in class 11 NCERT. Okay, another one is in biotechnology point of view in class 12 biotechnology lesson. Okay, so divide this plasmid MCQs into 2 sections. Both are important. Fine. Next, plasmid MCQ. You have the code words here: extra chromosomal DNA, autonomous replication. What are the code words used? Then third is phenotypic character. Which one gives phenotypic character to the bacterial cell? The answer is plasmid. They can ask it as a physical trait or antibiotic resistance. Next, in biotechnology point of view, they can ask it as cloning vector, selectable marker. Okay, then it can be also asked in the structural appearance way. Okay, small circular naked DNA means plasmid. Now, what are plasmids? The question is, what are plasmids? Let's see what are plasmids. Do you know any idea about plasmids? What are plasmids? If you know the answer, type it in the comment section. See, they have mentioned here extra chromosomal DNA. What does this mean? Some extra chromosomal. This, what is this attachment extra means? Very simple, easy. You have a bacterial cell, rod shaped bacteria. Okay, bacterial cell. Now, in you, this is a prokaryotic cell. Tell me now, do the prokaryotic cell have nucleus? Does it have nucleus? No. But in eukaryotic cell, you have well developed nucleus, isn't it? So here the nucleus is absent. Then where is the genetic information present in the prokaryotic cell, in bacterial cell? It is present openly in the cytoplasm. Okay, it is present somewhere in the cytoplasm. It is a small, it is a circular DNA. I am not going to use the word small. This is the genomic DNA. Okay, you have to understand this. First, I am going to talk about only this. Here you have nucleus. In the eukaryotic cell, you have nucleus. In prokaryotic cell, there is no nucleus. But still, it has to carry the genetic information, right? Genetic information means what? You have to keep asking the question, what is the meaning of genetic information, students? Doctors, type it. Blueprint of the cell. The information is in the coded word. It is passed from generation to generation. From one generation to another generation. This is the one which decides everything. Okay, the genetic blueprint. So where is it present in the bacteria? It is present openly in the cytoplasm, and we call this as genomic DNA because it carries genes, isn't it? Genomic DNA. Now this is the one which is equal to nucleus of the eukaryotic cell. This region, you call it as the place where this genomic DNA is present, is called as nucleoid. Nucleoid means something which looks like, it is nucleus like, but it is not actually a nucleus because it doesn't have nuclear membrane. Okay, it is simply a genetic information present openly in the cytoplasm. Fine. Now other than this, there is a small circular DNA. This is, the word small is important. This is bigger in size than this DNA. Okay, small circular DNA, that is extra chromosomal. This is chromosomal DNA. This is extra chromosomal DNA, which is called as plasmid. Do you understand? This is chromosomal DNA. This is extra chromosomal DNA, and this extra chromosomal DNA is called as plasmid, which is seen in the prokaryotic cell, an important characteristic of the bacterial cell. Because in biotechnology we use bacteria to prepare what? What type of hormone is prepared? And what is the organism used in that? If you know the answer, type it in the comment section. Now, I think you understood the meaning of extra chromosomal DNA. So, when you see the word extra chromosomal DNA, the answer is plasmid. The answer is plasmid. Next, autonomous replication. I told you, see here you have genomic DNA, that is the genetic DNA, okay, genomic DNA. Here you have plasmid. This will replicate, that is multiply on its own. It does not depend upon this DNA for multiplication. That is called as autonomous replication. It will not wait for this DNA to divide or multiply. It will multiply on its own, okay, independently and independently and autonomously. Okay, it can copy many times, that is multiply many times. Okay, multiply or copy many times. So the second characteristic is what? Autonomous replication. If you see the word autonomous replication in the sentence, look for immediately plasmid. Okay, immediately you have to look for what? Plasmid. This one is very important in biotechnology, origin of replication. Okay, this we will study in the biotechnology lessons. Next, the third is the phenotypic character. What is the meaning of phenotypic character? Expression, physical trait, okay, expressing outside, expressing outward, which is helpful in antibiotic resistance. These plasmids help the bacteria to face the antibiotic with a bulletproof. What does the bulletproof do? For example, you are going on a rainy day, you wear a raincoat. What does that mean? You get extra protection. You get extra protection by adding a layer of raincoat, isn't it? Same way, this phenotypic character means they are the traits which is expressed by the plasmid to protect the bacteria, which increases the survival of the bacteria, and this is one example of this is antibiotic resistance, which is given in your NCERT, antibiotic resistance. So phenotypic character, physical trait, example is antibiotic resistance. Look for these three code words for plasmid. Next, it acts as a cloning vector, molecular vehicle. Okay, this is in lesson biotechnology class 12. What is the meaning of vector? Type it in the comment section. What is the meaning of vector? Vector means, see, I am carrying this pen from here to here. Now, which is the vector here? My hand. So, vector is the one which is used to carry. Do you know one insect which is a vector? Do you know the answer? It comes in your class 12 lesson. If you know the answer, type it in the comment section. The insect which acts as a vector. Okay. So, these plasmids acts as a cloning vector, which is a motor vehicle. Okay, next it acts as a selectable marker. This can be used asked in the match the following. This too I will explain when we cover biotechnology code words. Okay, just to cover it along with the plasmid I have mentioned here. Today I am going to cover only the bacterial part of plasmid, class 11 plasmid alone. Okay, but still you can by heart this too and keep it ready. Next is this small circular naked DNA. If they used these words, small circular naked DNA, then it refers to plasmid. You should not get confused. Okay, there is a see, I told you here it is also circular DNA. Here also circular DNA. How we are going to differentiate which one? The word small is the one which gives you a clue. Okay, if it is small circular DNA means you have to search for plasmid in the option. So it can be asked as small circular naked DNA which is seen in the prokaryotic cell. You call it as plasmid. Now, genomic DNA versus extra chromosomal DNA. Right now I explained, in the cell you have the DNA. This is the genomic DNA. Genomic DNA. Okay. This is the circular, small circular DNA, which is the extra chromosomal DNA. What is also called as? Type the answer in the comment section, students. It is also called as plasmid. Okay. Now we are going to see the difference between genomic DNA and extra chromosomal DNA. Suppose if I am going to take this out of the bacterial cell, what will happen to the bacteria? Nothing will happen. It can survive. Suppose if I am going to take this genomic DNA, the bacteria cannot survive because here only all the command will pass to the cytoplasm and other structures. This is the one which governs the metabolic activities of the cell. So this DNA is very, very important for the bacteria to stay alive. Okay, and then this is the one which carries the blueprint of the bacterial cell. Now shall we see the comparison? This genomic DNA is the blueprint of the bacterial cell which carries the genetic information of the bacterial cell. This extra chromosomal DNA is outside the genomic DNA. That is why you call it as extra chromosomal DNA. These are also code words. Okay, outside the genomic DNA. This is a genomic DNA outside the genomic DNA. Okay. Next, this is a circular chromosome. Single chromosome, circular DNA. That region is called as nucleoid. Here you have to remember there is no nuclear membrane. All those complicated structure and organelles are absent in prokaryotic cell because they live a very basic lifestyle. No compartmentalization, simple lifestyle. Okay, prokaryotes follow minimalism. Easy to remember. You all know about minimalism. Do you know about minimalism? If you keep everything minimal, it will be easy for us to remember and maintain. So even your mental health. Okay, so minimalism. Prokaryotes have minimalistic lifestyle. Okay, next, this single chromosome. Here it is small, word small. Remember the word small, circular DNA. Here it is important for the mandatory for the cell to stay alive, as I said, because it carries the genetic information. Next, here this is the survival. This one gives you the survival perk. That is, I told you, like bulletproof. To face a hard situation, a tough situation. I'll tell you one. I'll ask you one question. Find it out and comment me in the comment section. Even after the live is ended, there is one bacteria which is in a different form. Okay, the form is different. It can be it can stay alive in a spore form. Okay, spore form. After any injury, particularly road traffic accident, you get the TT, that is tetanus toxoid. Now identify the bacteria. What is the importance of spore form and the tetanus toxoid? Okay, if you know any one of this, type it in the comment section even after the live ended. Okay, this is very important medical point of view. Even you might have seen your family members or someone, your friends, they immediately after they get an injury, they go for TT, particularly after a road traffic accident. Why do you do that? What is the reason for that? So if you know the answer, type it in the comment section. Even after that, I want you to find the mechanism, the pathophysiology behind this, and type it in the comment section. Now, survival perks means it acts as an extra shield for the bacteria, that is by expressing phenotypic character, which is the antibiotic resistance. Okay, so how do we find antibiotic resistance? There is something called as culture sensitivity. Find out what is this. This is out of Neet, NCRT, but still very interesting. See, if you correlate one with the another, it is easy for you to remember. This is called as memory hook. You call it as memory hook. Okay, then to monitor bacterial transformation with the foreign DNA. This is biotechnology part. What are the two uses of plasmids? Antibiotic resistance and monitor bacterial transformation with the foreign DNA. Okay, now shall we revise it once more? Shall we revise it? That is block 1. We have scientist and discovery, Golgi apparatus in the year 1898 by Camillo Golgi. It can be asked as a reticular structure and densely stained structure which is close to the nucleus. So be thorough with this. Then ribosomes, they are also called as granular structures, or they can be given as pellide particles. In the year 1953, discovered by George Pellade. Next you have plasma membrane or cell membrane, the selectively permeable membrane. The nature of this membrane was established by Singer and Nicholson in the year 1972 as fluid mosaic model or quasi-fluid membrane, which means in the ocean of lipids, the proteins float laterally, lateral movement. Okay, we have seen 4 scientists and discoveries. 4 scientists. Next you have plasmids. In the plasmids, we saw about the code words. Okay, today we saw about the bacterial importance. Biotechnology also you have to study. If you are in class 12, study about plasmids. Okay. Next, what are the code words given by NTA? It is called as extra chromosomal DNA, autonomous replication. ORI means origin of replication. Next, the third one is phenotypic character, that is the physical trait which can be expressed as antibiotic resistance. Then the fourth one is cloning vector, motor vehicle, both means plasmid. Look for plasmid in the option. Then it is a selectable marker. It can be asked as a small circular naked DNA. Okay. These are the 6 code words which is used by NTA to ask about plasmid. Next you have genomic DNA and extra chromosomal DNA. The difference between the two. This is the genetic blueprint of the cell. This is the outside genomic DNA. That is why it is called as extra chromosomal DNA. This one is a single chromosome, circular DNA. The region is called as nucleoid. It is very important for the cell's life. Here it is the small circular DNA which will enhance the survival efficiency of the bacterial cell in the way of providing extra phenotypic character like antibiotic resistance, and it is also used to monitor bacterial transformation with foreign DNA. So students, how was the session? Did you enjoy? How much you understood? If you didn't get any point, if you want me to explain anything in detail, you can type it in the comment section. I'll do it as a video. This is the code words session, so we are going to write only the code words. This half-an-hour series is to study only the code words. This will be useful for you in the last minute of the exam, the revision. I told you to make a bundle of white sheets and start writing it from today, so that at the last minute you can easily revise it. Okay, you need not go through the complete NCERT theory. You will not have time. So otherwise you can stick this in your study table area and you can see it daily. So by looking at that, your mind will automatically recall it. Okay, so students, have a break time after this, and later on practice physics and chemistry. I'll meet you in tomorrow's session. Take care. Bye.